So river blindness is it's caused by Onchocerca volvulus. It's a parasitic worm, and it is transmitted by black flies. So, and these black flies will breed in river rapids. So that is how it gets its common name, river blindness. So you can go. Let's go to the next one. You can see the image of an adult worm. The adult worm gives birth to baby worms here. These are microfilary, and then a file image of what was a river rapid, and also a file image of a black fly. Next slide, please. So what are the clinical manifestations of river blindness? So the clinical manifestations are usually skin, and then they occur in, please let's go back. They occur in skin and then in the eye. So we have 20 million people infected with volvulus, and out of this 14.6 million with a range of skin diseases. And they have about 1.1 million with blindness. And 99% of the infected people are in Africa, 31 African countries, with a few foci in Latin America and Yemen. Next slide. So for the control, to be able to have an effective control of the disease, you need to understand the life cycle. So there is an important stage, a stage where a black fly, which is infected with the infective stage, the third stage will take a blood meal on a human body and a human being, and then pass on the infective stage onto the human body, and then it goes through several modes, and then there's that ends in the adult, and then adult male and female. So there's a sexual reproduction that will lead to live baby worms that are also then passed on to the next black fly on the blood meal. So this is a cycle, and having this picture in mind is very important in the control of this disease. Next slide, please. So next one, next. Yes, so one control technique will be to surgically remove the adult worm and females in the noodles, and this is quite intrusive, but it's also it could be an effective way of controlling river blindness. Let's go next one, please. Also, the mass administration of ivermectin will kill the baby worms and prevent intake by the black flies, and also has a contraceptive effect on the adult female. Next. So the vector control will kill the black flies, will prevent the black flies from passing on infective stage, and also prevent them from taking on the microfilary during the blood meal. Next slide. In all this, diagnosis is very important. In the control of onco, the diagnosis is very important. So there are various forms of diagnosis. There's microscopy, there's PCR or QPC, real-time quantitative PCR, and there's also the lamp. Let's go to next please. And all these things are very important. They are very important, but these assays are either qualitative or quantitative. So you have it's either qualitative or quantitative. But we are asking ourselves, what about thinking or using an assay that can be qualitative, quantitative at the same time, and also provide information on the phylogeography of worms and also of the black flies that are involved in this disease. Next slide, please. So this is when we talk about the MinION sequencer. So next please. So this is how the MinION sequencer looks, and this is against a regular big ballpoint. You can see the size of the sequencer. This is the size of the flow cell, and also this is a sequencer that is connected to a computer. So this is how it looks, and to make it clear to you, we have put it by a ballpoint so that you can appreciate the size very well. Okay, next slide, please. So as brief introduction or brief background, the MinION sequencer has been used by the University of Birmingham for the control of Ebola virus in Africa and Zika virus in Latin America. So there's already a proven record here. And actually, to go ahead of that, I will say that the validation happened in the same university, the University of Birmingham. So actually, the aim of this project was to use the knowledge and then the experience of the University of Birmingham to validate a MinION sequencer as a tool to help fight river blindness in Africa. So next slide, please. So as prior to the validation exercise, prior to sequencing, sampling was done in some key areas in Ghana. So we sampled black flies and also next slide, and also the worms. And this was done by the lab team from the Council for Scientific and Industrial Research and also the Noguchi Memorial Institute for Medical Research. Next slide. Now the experiment, let's go next slide, please. So this, as I said, the validation happened in Professor John Corbon's laboratory in the University of Birmingham, and I ran you through the workflow. So the first stage, this is the Cas9-mediated sequencing. The first stage was to block the ends by removing phosphate groups. So you block the ends of the DNA by removing phosphate group. Then after you removed, no, let's go back, please. After you remove the phosphate groups, now you introduce the Cas9 RNA or Cas9 RNA. So this Cas9 were targeting the mitochondrial genomes of the black flies and also the worms. So they would cut specifically out your region of interest. So you can see the Cas9 here cutting this region of interest. And after cutting, what happens is that it now exposes sticky ends because now all other regions have been blocked. And those sticky ends are now A-tailed, so we add A-tailing, and then the adapter, that is the MinION sequencing adapter, is added to the A-tail. So what happens is that you have DNA that is extracted from the whole genome, but because of the because Cas9 will target your region of interest, and all other regions are blocked, it is only the region that has the abductor that will be sequenced. So by this approach, we were able to sequence the mitochondrial genome of the black flies and also of the worms. Next slide. So next, so we use four Cas9 guide RNAs for both worms and the black flies, and we screen them using equimolar concentrations of DNA, and also we varied a lot of reagents and then conditions. And we discovered that for both the worm and then the black fly, we had one of the guide RNAs produced significantly higher yields. Let's go to the next slide, please. Next, please. Next one. Next, please. So this is the map. This is a mapping of. It's a picture of a mapping of Cas9-mediated sequencing against another experiment that we did. So this one, the whole genome sequencing was when we didn't do any end blocking, but after just the DNA, and then we added sequencing adapters to the DNA. So this one was not targeted, whilst the Cas9 was targeted. And you can see deep sequencing for the Cas9 as compared to the whole genome. Next slide. This one also shows you the length of reads, the length of reads that map to the mitochondrial genome. This is for the whole genome sequence. You can see the largest read length was around 5,000 base pairs. Next, please. For the Cas9, please, no, let's go back, please. Let's go back, please. For Cas9, when you look at the read length that maps to the mitochondrial genome, we have you have one read that is almost the size of the mitochondrial genome. That's about 12 kb. So you can see that in a single run, you are able to almost sequence the whole mitochondrial genome of an organism. So we have one read that's about 12 kb, and just in one run. Next slide. So what we did is do some phylogenomics. So we got some sequences, whole genome, mitochondrial genome sequences from Prof. Charles Brockhouse and then Prof. John Coburn. And so these are whole genomes that were from African species and also from Asian Similon species and European species. And this up here is our sequence. And what you can see from this phylogenomics is that you can see the sequences from our run aggregating with the African species up here. So this is just a graphical view of, you can see the sequences aggregating and showing the importance or showing that this sequencer can be used to in phylogenomics. Let's go next, please. So as future works, we want to have a proof of concept that this sequencer can be used for in the fight against river blindness. We want to also work more and even make it even more sensitive and specific. We also want to perform more trials on different black fly and worm samples from a wide range of endemic communities in Ghana. And also we want to document our findings in publications. And we look eventually to have this platform adopted by control programs. We also want to look for funds to continue the work we are doing. So the research team that did this validation already applied for a FLAIR fellowship from the Royal Society. We were not successful, but that will not stop us. We want to look at other opportunities to look for funds to continue this work. Next slide. So a quick one here. So on my return from the training, that was in early 2020, I came back to Council for Scientific and Industrial Research. And just around that time, we had the COVID-19 outbreak, the first cases in Ghana. So the president of the country called for the increase in testing facilities. And because of the importance of the Council for Scientific and Industrial Research in science in Ghana, we didn't have any option but to be part of the testing facility. So we have a PCR testing facility, and I'm the team leader there, and then another team member, Professor Osar Kenebona, is the principal investigator there. So as it stands now, we are doing COVID-19 PCR tests here, but also the point I want to make here is that we have a lot of samples that we want to start using the knowledge from the MinION sequencer to do to deep sequence. So you want to use the MinION sequencer to be able to help in tracking various variants or various viral variants, just like the University of Birmingham did with Zika and Ebola. We want to replicate that here using the SARS-CoV-2 virus. Next slide, please. So these are publications of the work that we've done, and these are protocols that have been published on the Gnatwork website. So the links are here. You can check these protocols for more information. Let's go, please. Next slide. So next, please. Our summary slide. So we are saying that the Cas9-mediated MinION sequencing protocol has been validated using samples collected from Ghana. The protocols have been published on the Gnatwork website. Next, please. We want to look for more money to expand this work, so we are going to write more proposals, and we're going to search for grants to do that. And as I said, we want to use the knowledge gained from this project to sequence SARS-CoV-2 genome. Since we are now a COVID testing facility, we have the sample, so we can do that. Next slide. I'm very grateful to this team. This is a very important slide. Professor John Coburn has been an amazing mentor and leader. He and his team were very, very instrumental in instrumental in passing on this knowledge, and they made my visits very, very worthwhile. So I really appreciate them. Professor Charles Brockhouse has always been shown keen interest in the work that we are doing, always passing on his technical knowledge. So really, really appreciate him. Professor Michael Sayachinebwana is my boss in Ghana, and he has been very, very instrumental in my growth, always watching, making sure I'm on the right path. And then Francis Verrier, my colleague here, Professor Boachi has also been very important and always watching me, making sure that I'm on the right path and a key mentor in my life. So I appreciate all these key people, and also Gnatwork and then the Playwright Institute and all other funders that were involved in this project.